Thank you very much for joining us today to share your knowledge with the audience about endometrium and the latest updates available on the subject, as well as talking on reproductive tract microbiota. I am thankful to you and I welcome you on behalf of the audience, on behalf of Japan, Middle East, and Indian team of Igenomix for sparing your time and coming here, and you will do your best to enlighten the audience, I'm sure. Dr. Emma, before I hand over the session to you, I would just give a brief introduction of yours to the audience.
Okay.
And then the session would be all yours. Please permit me to go ahead. Dr. Enmaculada Moreno is a research co-director with Igenomix. She graduated in biochemical sciences in 2002 and obtained her European PhD in 2007 in the field of molecular biology, which was awarded the best PhD thesis of University of Valencia, Spain in 2008. During her postdoctoral training, she specialized on molecular oncology and regenerative medicine. In 2014, Dr. Moreno joined the R&D department of Igenomix, where she is currently the research co-director and leads the research on the role of the microbiome in human production. From 2015 to 2018, she collaborated with reproductive and stem cell biology laboratory at Stanford University School of Medicine as a visiting scholar. And currently, Dr. Emma is settled in Boston and manages the new research lab of Igenomix in the United States. Dr. Moreno has been productive in both basic and clinical research. She's the author of more than 30 international publications in peer-reviewed journals and has participated as invited speaker in over 50 international conferences. It's wonderful and very impressive of you, Dr. Emma, and I hand over the session to you. It's all yours.
Thank you, Vikram, for your very nice introduction. It is my pleasure to be here and share this piece of work with all of you and all the attendants. So good afternoon, good evening to everybody, wherever you are. I'm going to talk today, as Vikram has already said, about the latest updates of the endometrial factor, and I'm going to focus about the reproductive tract microbiota, which has been the main focus of my research in the last, let's say, 6 years. So this is a picture of the first, what we call the first step of life journey. And as you can see, we have two main actors, two main players playing in this field. We have the embryo, which is of course, super important for the pregnancy to start, and we have the maternal endometrium. And in my talk, I'm going to focus about the endometrium, and I will try to let you know which have been the advances. You know that in Igenomix we quite like in Igenomix Foundation to study the endometrium, and I would like to start a little bit about talking about the clinical relevance of the endometrial factor. As we said, there are at least two factors that are playing an important role in the pregnancy, in the starting of the pregnancy. The first one is the embryo, and I know that you had Dr. Carmen Rubio 2 weeks ago, and she's one of the leading experts in the field in embryo and embryoneuploidies, and we have also the endometrium. Regarding the embryo, you know that we as humans, we are very inefficient, unlike other species, to produce good quality or normal chromosomally embryos. And for example, compared to mouse, where the embryoneuploidy rate is very, very low, in humans we have between 40 to 100% of embryoneuploidies, depending on many factors. One of them is the age of the mother. However, this makes that because our embryos are, let's say, less quality, this affects the implantation rates. For example, in rodents and rabbits, we have an implantation rate that is close to 100%, while in humans, in a natural cycle, is close to 35%. And this cannot be only the consequence of embryoneuploidies, because If we transfer human euploids, human euploid embryos, we know that the implantation rate is close to 60%. So there's still a lot of room for improvement in this way, apart from working on selecting embryos for implantation. It is more or less considered that the endometrial factor affects to 30% of infertile couples. And within this 30%, there are many different factors that may play a role. In Igenomix Foundation and in Igenomix, we have been working for a very, very long time in the synchronization between the embryo and the mother and the endometrium, and how this may improve the implantation rates and the pregnancy outcomes. And this is the basis, the rationale for our ERA test. And there are also other, this is regarding the endometrial receptivity, but there is also other factors that may play a role. For example, the maternal embryonic crosstalk, which are the signals that the endometrium is producing and sending to the embryo to say, hey, hello, I'm ready to implant. Are you ready to come and invade? And the other way around, also the decidualization, you know, that first, the embryo has to adhere to the endometrium and then has to invade in the decidualized stroma. So if there is a defect in the decidualization, this may produce long-term issues during the pregnancy, like, for example, preeclampsia and so on. So there are many different roles that the endometrium is playing in pregnancy, in both in early pregnancy or preconceptional and periconceptional moment, and also during the pregnancy. But today I'm going to focus about the microbiome. And why the microbiome? Why is now this hot topic? So the microbiota, as you know, microbiology was a very classic field of science that was, let's say that we forgot about it many years ago, because in the developed countries, the infections and the microbiological problems are more or less managed clinically, and they do not produce big issues. And for a long time, the microbiome has been considered that it was more or less controlled by the clinicians, and we didn't pay much attention to it. But in the last decade, we have been revolutionizing this field of the molecular microbiome, because we have learned that we do not only have pathogens in our body when we are sick, when we have an infection. We know now that apart from our human cells in our body, we are full of microbiota. This microbiota is comprised of mainly bacteria, but also yeast and also virus and many other species. And these bacteria are not only pathogens that may put into risk our health. They are living with us, and they are producing some of the functions, the physiological functions in our bodies. For example, the synthesis of vitamins, the training of the immune system, helping with digestion of the nutrients of the food. So they are not only these bad guys that are putting our health into risk. So in this scenario, we know now that the microbiota, the number of bacteria, for example, is, let's say, at least the same number. We have in our bodies the same number of bacteria than human cells, and much more genes that are expressed from bacteria than from human genes. For example, the bacteria in our bodies are expressing approximately 9 million genes, while our body, our human cells, are only able to express 23,000 genes. So in this context, we think that analyzing the microbiota and analyzing the interactions between the host, we, the human, and the microbiota, which is very, very important, and these potential disbalances in this microbiota is what can put us into a disease. If we go to analyze the urogenital tract microbiota, what we know, and this is the work of a human microbiome project, we know that the urogenital microbiota is mainly composed of Lactobacillus species. But also we can find other species, like, for example, Gardnerella, Atopobium, Isniatia, Bifidobacterium, some other bacteria that can also be there, although we found them in a lower proportion than Lactobacillus. And Lactobacillus has been consider that is the bacteria that all the, let's say, women should have in a healthy status. But there are many influences between the bacteria in our genital tract and that may produce this eubiosis, this normal state or this healthy state, and the dysbiosis or these pathogenic states. And the microbiota may be influenced by different factors. For example, the hormonal status during the menstrual cycle, sexual activity, of course, any episode of infection because of exposure. So there are many different factors that may affect the microbiota in the vagina. And in turn, these changes in the microbiota may lead to other types of diseases or conditions, like, for example, bacterial vaginosis, infertility, and so on. So, which is really the impact of this vaginal microbiota in infertility and obstetrical complications? So we know, as I said before, that several studies have associated low lactobacillus content in the vagina with poor reproductive outcomes. So what we consider a dysbiosis, or let's say a pathological state, will be a decrease in lactobacillus in the vagina. Which is the risk of this? So, for example, some pathogenic bacteria, for example, Neisseria gonorrhea, Chlamydia trachomatis, and some mycoplasma species, we know that they are direct causes of infertility. So, for example, all these bacteria that are transmitted through STIs, through sexual transmitted infection, is going to cause infertility directly. The prevalence of having an altered or an abnormal vaginal microbiota is around 20 to 30% in asymptomatic women in the healthy, let's say, healthy population, and this percentage goes up to 40% in patients that are undergoing IVF. So, for example, in our clinics, even if the patients are asymptomatic, they do not feel any pain, any itch or whatever, approximately 40% of the patients that are attending our clinics are going to present some dysbiosis in their vaginal microbiota. And the consequences of this dysbiosis, for example, if we focus in the vagina and the bacterial vaginosis, which is an alteration of the vaginal microbiota, we know that this is responsible for an increased risk of miscarriage, either early miscarriage or late miscarriage, also increased risk of premature rupture of membranes and preterm labor. But we are focusing reproduction, and at the end, implantation occurs in the endometrium, in the uterus. It does not occur in the vagina. So some years ago, we asked ourselves what about the endometrial microbiome? Is there an impact? Is there an endometrial microbiome? And if so, is there an impact of this endometrial microbiota in reproduction? So this is a slide that summarizes more or less the knowledge that we have been acquiring about the uterine cavity sterility. And if you see in this graph, from the beginning of the 20th century up to more or less half of, or a little bit more than half of the 20th century, we all thought that the uterus was sterile. And this is because, as I mentioned in the introduction of this talk, we have always considered the microbiota as something that is bad for us, that is causing an infection. And nobody could thought about, at that time, about a new life being created in a contaminated environment. Then from 1960, more or less, some researchers started to study this using microbial culture. They were getting samples from the endometrium, or then afterwards using the transfer catheter when they started doing IVF. And they were analyzing these samples by doing microbial culture. And what they found is that there were, in some cases, they were able to isolate bacteria from the uterine cavity of the woman that they analyzed. But there was a little bit of controversy at that time, because some studies say that yes, there was an endometrial microbiota. There were other studies saying that no, there's not. The microbiota is sterile in the uterus. And from 2007, in the last, let's say, 10 to 12 years, every study that has been considering the uterine cavity microbiota have found bacterial DNA in there. And why this change in these 3 periods, coming from the no, there's not, maybe there's one, to yes, we know there's one. So the difference is the technology that we are using. For example, in the period between 1957 and 2007 and 2006, the researchers were using microbial culture, and we know that more or less 60% of the bacteria that grow in our bodies are not easy to culture in vitro, and they do not grow in vitro, or they need very specific conditions to grow in vitro. So sometimes when they say this culture is negative, it's not because there was no bacteria in there, but the bacteria maybe were not easy to culture, or it was not, the conditions that they were using were not proper for this type of bacteria. Now, from 2007, we started to study microbiology from the molecular point of view. So we started to analyze the nucleic acids of the bacteria in the samples. And this technique, the NGS, is next generation sequencing, is much more sensible, and it allows us to detect very small amount of bacteria and with a lot of resolution. So that's why we are now able to analyze the bacteria in some niches, not only the uterine cavity, but also in some other niches that we thought that they were sterile before. If we go to analyze which is the relation in the literature between the endometrial pathogens and the human reproduction, there is a collection. This is some examples of the published papers in this regard, and these were all papers in which the researchers were analyzing by microbial culture the tip of the embryo catheter after the embryo transfer. And what they found is that when they were able to isolate this list of pathogens that you have in the slide, they showed that there was a decreased implantation and pregnancy rates in their patients and increased miscarriage rates. So these works pointed out to the hypothesis that the pathogens in the uterine cavity at the time of embryo transfer may produce a negative effect on pregnancy rates and pregnancy outcomes. So some years ago, we started our research, and we tried to translate all this knowledge into molecular microbiology with the objective to really confirm whether there was an endometrial microbiota and the potential impact of this endometrial microbiota in human reproduction. And this was the paper that we published back in 2016 with the results of this research. So we use this technique. This is using NGS, next generation sequencing. What we do is we obtain the samples, either endometrial or vaginal aspiration. We purify the DNA in these samples, and we make use of this gene, which is the 16S ribosomal RNA gene of bacteria. This gene is very special because it is a gene that is conserved in all the bacteria. So every bacteria in the world has this gene and has some pieces of the gene, some sequences of the genes are constant, and some of them have evolved through the evolution. So the sequences that you have now here, picture color in red, they are variable, so they are specific of each bacterial species. So we can amplify these bacterial sequences, and then we can annotate which bacteria each one and which is the relative abundance that we have in them between among them in our sample. So using this method, we compare the endometrial and vaginal microbiota of fertile and asymptomatic women, and this is in this graph here. You have the average amount of bacteria that we found. For example, in blue, you have the percentage of Lactobacillus. And as you can see, the content of the microbiota in endometrium and vagina are, in average, quite similar, and they are mainly composed of Lactobacillus species, as expected. However, if we analyze each pair of samples from each woman, if we analyze for each of the women that we tested the endometrium and the vagina, and we compare this, we found that in some women, approximately 20% of these fertile women, there were some differences between the bacterial composition in the endometrium and the bacterial composition in the vagina. And this prompted us to continue with our study and analyze the endometrial microbiota in relation with the reproductive outcomes in those women undergoing IVF. Our results were later confirmed by this work of Chen and collaborators that was published in Nature Communications, and they explored the microbiota throughout the reproductive tract, collecting samples from the vagina, from the cervix, the uterus, the endometrium, and also the fallopian tubes and the peritoneal cavity. And they found that Lactobacillus there was a gradient, there was a continuum in both the amount of bacteria that were found in the microbiota of the reproductive tract and also in the type of bacteria. And what they found is that Lactobacillus was the most abundant in the vagina. However, as you go up in the tract, the amount of Lactobacillus is getting lower. And the same with the total amount of bacteria. They say that the amount of bacteria in the endometrium, in the uterine cavity, is between 100 and 10,000 less than in the vagina. So it's a low biomass microbiota, but still consider that Lactobacillus is the majority genus in these samples. So for our study, we continue our study using this design. Now that we have analyzed the endometrium and the vagina microbiota, we focus on the endometrial microbiota in women undergoing IVF. So the design of our study was, we started with 35 recurrent implantation failure patients for this pilot study. And in these patients, we collected an endometrial fluid sample just before the endometrial sample for the ERA test. So in this case, what we did is we did the ERA test, we transferred the embryo, synchronizing the embryo and the endometrium. So we did personalized embryo transfer, and then we follow the pregnancies of this woman. And at the end of the study, this was a pilot observational study. What we did is to compare the live birth rates with the microbiota that we analyzed in the endometrial fluid. And these are the results of our pilot study. As you can see here in this heat map, this is each of the rows in this graph is the main bacteria that we detected in our samples, and each of the columns corresponds to one of the patients. And what we saw here is that there are some of the bacteria that are very, very highly expressed, the ones that have the red color. So, for example, Lactobacillus, Streptococcus, Bifidobacterium, and Gardnerella had a high percentage, a high abundance in some of the samples. So we asked ourselves whether it was possible to predict the reproductive outcomes of these patients based on this, the abundance of these bacteria. And what we saw is that using two mathematical methods, the answer was the same. And it was that the percentage of Lactobacillus with a cutoff of 90% was predictive of the reproductive outcomes of these patients. So we went back to our results and we classified our samples, our patients, into those having more than 90% of Lactobacillus, and we call this group Lactobacillus-dominated microbiota, and those with less than 90% of Lactobacillus, and we call this group non-Lactobacillus-dominated microbiota. And then we compare the results with the actual results that they had in their IVF treatments. And for example, if you see here, you have patients classified by those with a higher than 90 Lactobacillus-dominated microbiota, lower non-Lactobacillus dominated microbiota. And if you go to the pregnancy, implantation, ongoing rates, etc., you may see that those patients with lower Lactobacillus abundance and, of course, higher number of dysbiotics or pathogens had lower pregnancy rates, implantation rates, and live birth rates per transfer compared to those with Lactobacillus-dominated microbiota. So it seems that this is confirming our previous hypothesis. This can be also seen here in this graph, in which I have split the patients by the reproductive outcomes. So here you have those that had a live birth on the left, in the middle those that had a miscarriage, on the right those that were not pregnant after the personalized embryo transfer. And if you can see, those with a live birth presented a higher amount of lactobacillus, which is depicted in blue color, compared to those with miscarriage and no pregnancy. And on the other side, those with a miscarriage or no pregnancy presented higher amount of, for example, Gardnerella or Streptococcus or Veillonella and other type of bacteria that have been considered dysbiotics or pathogens of the reproductive tract. Then, once we have finished this pilot study, we underwent a prospective study that I will show you at the end of the presentation. But in the meantime, we started to analyze some of the cases that we started to see more in depth, because we start to find specific cases, clinical cases that really caught our attention. This is one of them that I would like to present. It was published at the end of 2019. And this is the case of a woman that attended the clinic with primary infertility. And this is the follow-up. I am not going to go in deep into the clinical case, but to summarize, this is a follow-up for 18 months of this woman that in which in these 18 months we had 6 different samples to study the microbiota, and she had 3 different embryo transfers. We did this because it is very difficult to sample the endometrium of a woman longitudinally. So this is, to my knowledge, the only work so far in which the same woman has been tested 6 different times throughout the time. So I'm going to present you the results. These are the 6 different samples that we analyzed. 3 of them, as you can see here, number 1, number 3, and number 6, were associated to or were collected prior to an embryo transfer, and these are the results of the transfer. The first one, she had an ectopic. In the second one and the third one, she had miscarriages. And as you can see, one of the main bacteria in her reproductive tract was Gardnerella vaginalis, which is one of the most fastidious pathogens in the reproductive tract, and also in not only in the vagina, but also in the endometrium. So we really were very concerned about this case, and we went a little bit in deeper into the study of this case. And we focused in this last sample because we collected the sample just 7 days before the embryo transfer was performed. So there was no chance that the microbiota was fluctuating between one cycle or the other. So we collected this sample only 7 days before the embryo transfer was performed, and we analyzed this sample not only for the type of bacteria that was present, but also at the functional level. And here you can see that this is the 16S RNA that I showed you before. But when we analyze the genes that are expressed, not only how many bacteria we can find, but which are the genes that are expressed in this environment, the majority of the genes were expressed by Gardnerella vaginalis. And when we analyze the functional status of this sample, we saw that the majority of the genes that we found were related to virulence factors and adaptation to the environment. So it seems that this environment, these needs in the uterine cavity of this woman was mainly focused in biofilm formation, in antimicrobial resistance, in cytotoxicity, and other virulence factors. I need to say that this woman, through all these 18 months, she had several antibiotic treatments, and we were completely unable to get rid of this Gardnerella, which is consistent with this antimicrobial resistance that we saw at the functional level. This is another case that was very curious, and it was in our hands. And this is the case of a woman that attended the clinic with primary infertility. And well, she had a first embryo transfer with euploid blastocyst. She did not become pregnant. Then, because she was a young woman, we suspected of the endometrial receptivity, and she performed the ERA test. And at the same time, we collected a sample from endometrial microbiota. Well, she had a personalized embryo transfer. She was pregnant but had spontaneous abortion, and then at 8 weeks of pregnancy, so she had accurates, and we analyzed the product of conception to confirm that the euploids that the blastocysts were euploid, and it was confirmed. So after the curates, she went home for a rest, and after the holiday break, she came back to the clinic, and the doctor decided to collect another endometrial fluid sample to analyze the endometrial microbiota before starting a new IVF cycle. So our surprise was that 2 weeks later, she called us and she said that she was pregnant. She had conceived spontaneously at home. And of course, she told us that when we collected this sample, this endometrial fluid sample, she was already pregnant. She was a 4-week pregnancy. Nobody knew. Of course, she didn't know at that time. The doctor didn't know at that time. So we collected the samples. We kept the sample, and once she had her baby boy, we analyzed this sample to check which were the difference between the microbiota before this abortion, this miscarriage, and during the early pregnancy in the same woman. And these are the difference. On the left-hand side, you have the microbiota of the miscarriage, and on the right, you have the microbiota of early pregnancy. And as you can see, the amount of Lactobacillus is very, very high, higher than 90% in the case of the pregnancy. However, before the miscarriage, she had a very low amount of Lactobacillus, it's lower than 20%. If we analyze this using also the species level, what we found is that Lactobacillus inner was the only bacteria that was present during the pregnancy. However, during the miscarriage, there were, apart from Lactobacillus, there were other types of bacteria present in the sample. But again, we were not only interested in which were the bacteria and in which percentage they were present in the sample, but also which were the genes that these bacteria were expressing. And well, this is a heat map showing that there are some genes that were expressed previous to miscarriage, and these genes are related mainly with transposases, with insertion elements, and this is somehow connected to genomic instability. However, during pregnancy, the genes that were expressed in this sample by the bacteria were, all of them came from Lactobacillus inners, and all of them were like very basic metabolic genes related to energy, to metabolism, and some of them also related to defense mechanisms. So clearly, the microbiota before the early miscarriage, before the spontaneous clinical miscarriage, and during the early pregnancy in the same woman was completely different. So the conclusions of all this work together is that the percentage of Lactobacillus together with the specific pathogens are significant variables to predict the reproductive success. Also that these results expand the evaluation of endometrial factor not only at the morphological and molecular levels, but also at the microbiological point of view. And of course, our observation led us to propose new diagnostic tools and personalized therapeutic procedures using molecular microbiology. This is our hypothesis and our concept. The better is your microbiota, the better are your chances to get pregnant, to have a successful pregnancy. And this has been the rationale for the creation of the development of these two tests, ENMA and analysis. And again, this confirms, I like very much this slide, and I always take this slide from the paper by Franasiak and Scott back in 2015 when we started this project and this research. And they at that time were, they foresee this implication of the microbiota in human reproduction, not only during the pregnancy, but starting from the creation of the gametes, through fertilization, implantation, during pregnancy, and even going into the neonatal life, so the first days of the newborn's life. And I just would like to show you some updates about what is next. So the work that we are performing now, I have shown you the pilot study, I have shown you the 2 clinical cases in which we have been focused in the last years. And now I would like to show you what we are doing. We are now immersed and we are finishing a prospective observational study in which our objective is to confirm our results of the pilot study and see whether there is, to confirm whether, to validate whether there is a relationship between the endometrial microbiota and the reproductive outcomes. So we undertook this prospective study in which we have analyzed, we have recruited 452 patients. These are IVF or ovum donation patients that have received personalized embryo transfer with blastocyst in a hormonal replacement therapy cycle. And these are the participant sites. We have selected clinics around the world to have a representation not only of our small 35 pilot study, but having big numbers and also a lot of diversity in terms of age, ethnicity, incomes, or whatever. So this is something that is very, very wide. We have women participant sites throughout the world in, as I said, in 13 participant sites in 8 different countries. The study is nearly finished. So we have the last patient was included in the study in February 2019. The last baby was born now in July 2020. And we are now doing the analysis, and I hope to have the results ready for publication very soon. And at that time, I will be able to share the results with you. So I just wanted to update you in what we are doing now and what will be the next results that you may expect from our side. So with this, I was just, I may finish my presentation. I just would like to thank all the people that have been involved in this research. Of course, Carlos Simon, which is the boss, the supervisor, and the one that allow us to perform all this type of research. Also the research group, especially to those that are involved in the Microbiota project, Marta and Yolanda, that are working hands-on with the samples, with the sequencing, and working really very, very hard. Also the bioinformatic team, Davide and Davide, and the people that is helping us with the clinical study, and the group of Carlos Gomez. And of course, our collaborators, all the centers that are participating in our studies and are helping us to recruit the patients and collect the samples, because without them, there is no chance to perform this research, and all the patients that have accepted to participate in our studies, in our research. Of course, I would also like to thank all the Igenomix family that is growing and is now worldwide, and all of you for your attention, and I'm open for questions if you have any. Thank you very much.
Oh, wonderful, Dr. Imma. Thank you very much. It's nothing is better than hearing from the one who has been, extensively involved in the research of these services, like microbiota or chronic endometritis, if we talk about. Thank you very much for this lovely presentation. I'm sure the audience must be sharing my views here with me, that this presentation was really wonderful, enlightening, and productive. As well, I should say, worthwhile spending your time on.
Thank you very much.
Yeah, thanks again, Dr. Imma. As you know, I'll start with the question and seek your answers. The first question comes from I should say, one of the pioneers in the field of infertility, Dr. Firuza in India. Her question is, does excessive lactobacillus impact on pregnancy?
Well, we are exploring now which is the functional role, because as I mentioned, we have been focused in the majority of research groups, and the majority of people that is working in the field is focused in how many lactobacillus you have, how many pathogens you have. But we think that the most important thing and the next step that we want to undertake is not only to consider the numbers, but also what are they doing there and how are they interacting with our body. So which is the connection or the interaction between the bacterial cells and the human cells. In the context of Lactobacillus, we sometimes think that maybe we don't know whether the role of Lactobacillus is to perform a positive role, or the role of Lactobacillus is just to avoid that other pathogenic bacteria colonize the needs. And this is something that we are going now to, for the next years, we are planning a big project and in which we are going to analyze at the functional level which is the impact, and we are going to do this through 3D in vitro models and also using single cell RNA-seq of patients colonized with Lactobacillus and with other type of bacteria. So about the functional role, we still have a lot to learn. And this is something that we will undertake from September on. We are going to start working in this regard. So the role of Lactobacillus is another bacteria is still under question. So we know that they are good, but we don't know why they are good.
Thank you very much, Dr. Emma. Another question is here, a couple of questions from Dr. Harsha. First question from her is, what are basically the causes of alteration of the microbiota in the endometrium?
Yeah, this is a very good question. And it seems, I mean, in the endometrium, it's what we consider is that the most plausible way of the uterine cavity colonization is the ascension of bacteria from the vagina. But there is still, we don't know which are the factors or how can you improve your microbiota in the reproductive tract. So there has been like many different hypotheses. There's people saying that genetically you have like a more risk of having some types of bacteria or others because your genetic factors makes you being more colonizable but one type of the other bacteria. Others points to hygiene, for example, or to sexual activity, to the use of different drugs or for example, probiotics, antibiotics, and so on. So it's sometimes difficult to know how can you shape this microbiota and which are the results of this. But the colonization of the endometrium, the colonization of the uterine cavity, we think that comes mainly from the vagina. And as I have shown you before, in the majority of the woman, the content of the vagina and the endometrium is quite similar. It's not identical in every woman, but in the majority of the woman is quite similar. So this will more or less give us an idea of what you have in the vagina may influence what you will have in the endometrium.
Thank you very much for this elaborate answer, Dr. Emma. Another question is, can the microbiota change from unfavorable to favorable type spontaneously in a woman?
We have seen many different cases. We have seen women that are very stable. So for example, women that have what we consider a healthy microbiota, and this is very stable throughout the time. We have seen women that, for example, the clinical case that I presented in which some women have some pathogens and they are all the time having different samples having the same pathogen, and whatever you do is not worth it. Even if you give antibiotics, probiotics, whatever, she is not going to change her microbiota. And also we have seen cases in which the microbiota may change from favorable to unfavorable and the other way around. So we have observed every case. So everything is possible. It can be spontaneously. Remember that every cycle we change our hormonal status, we have the menses, and then there is like a clearing of the endometrium. At least the surface of the endometrium is clear, and is again the endometrium proliferates again and is colonized again. So there's different cases, different scenario. Each woman is different, and that's why we consider that personalization of the reproductive medicine is important, because the thing that you apply to one patient may be different to the thing that you may apply to the other. So we consider that personalization. Unless you know which is the status of your patient, you cannot treat her properly.
Thanks again, Emma. There are various co-founders, like the protocol used during assessment cycle and may not be replicated in transfer cycle. Has that been addressed in your study?
Yeah, absolutely.
This question comes from Dr. Arvind. He also says, we now have data about the variation in the quantification of different Lactobacillus species in different populations. So how can the test customize to different populations?
Yeah, this is a very good question. And yeah, I will answer this in two different ways. Regarding the variability in the microbiota studies, we have two sources of variability. The first one is the clinical source. So, for example, as pointed out, the hormonal status in the sample collection and the comparison with the embryo transfer collection. If the hormones are impacting the microbiota in the reproductive tract, of course, you have to consider all the time the same hormonal status or the same type of therapy if you want to compare one cycle with the other. So this is very important. So the clinical cycle that you apply, if you are collecting the sample, if you are interested in the microbiota during an HRT cycle, you have to collect the sample in the same day of the HRT sample. And this is the way that we have been working in our studies. We always collect the sample during the window of implantation of the woman, even if it's in another different cycle, but always following the same protocol. So this is very, very important. This is, let's say, the biological or the clinical source of variability. The other one is the technical source of variability. And this depends on the type of sequencing that you are doing, and very important, it depends on the controls that you are using. And of course, you can find in the literature papers showing different results. What is very, very important is that every time that you read a paper or you try to understand the study that other people has been doing, is that to check whether they have analyzed blank controls and they have been consistently checking whether all the quality parameters are okay. Because we need to know that we are analyzing the microbiota at the molecular level. And even if you use sterile tubes, sterile water, sterile tips for the procedures, you may have bacterial DNA, even if this bacteria is dead, but you may have bacterial DNA that is going to interfere in your sample detection. So it is very important to analyze the blank controls, to have extraction controls, and to check for not only the clinical variability, but also the technical variability when you are working with this type of samples. This is very, very important, and is one of the biggest confounding factors in this field.
Thanks again. Dr. Alicia has a question. Do fertility drugs and hormonal treatment, gonadotrophin hormones, affect the microbiota? If we are triggering the patient with fertility drugs and gonadotrophin hormones, does it affect the microbiota? And if yes, up to what extent?
So we have not analyzed the microbiota during, for example, during stimulation cycles, because our main interest is during implantation. So we are interested in the window of implantation, in the time in which the embryo has to implant. So we have been focusing in this part of the cycle, but of course, I guess that there may be differences in the use of different hormones or different hormonal treatments. There is no published literature, to my knowledge, in this regard, but I'm sure that different hormones may impact at least For example, I have now a paper in my mind that is not about the endometrium, it's about the vaginal microbiota, and they show a graph in which they show how the stability of the microbiota depends or fluctuates regarding the moment of the cycle. And they show that the peaks of estradiol are the moments of the biggest stability, while the starting the proliferative phase of the cycle is much more variable. And of course, if you treat with different hormones or different drugs, you may, of course, make this happen. The other thing is not only the hormones that you use for the cycles, but also, and this is something that I consider that is quite important, at least in some countries, is the use of antibiotics that some doctors employ for the fertility treatment. Some doctors are abusing of the antibiotics during fertility treatment. They give antibiotics for every procedure. So this may also, of course, impact the microbiota composition at different times of the cycle.
We have many more questions, but I would like to take 2 or 3 more. Dr. Imma, during your research, have you found any identifiable difference between the microbiota in uterus and the microbiota in vagina?
Yes, absolutely. It's not in every woman, but in many of them. In fertile, healthy woman, I would say that is 20% following our results. And I guess that in fertile woman, the differences between the vaginal and the endometrial microbiota are bigger. We have found these differences, but we are not the only ones that have found these differences. There are other reports by other groups that have been studying, doing the same comparison. For example, the paper by Mitchell in American Journal in 2015, they correlate the vaginal and endometrial microbiota, and they found difference between the same patient in the 2 different niches. Also, the paper by Chen found these differences. So many other, and not only us, but other people have found changes in the differences. The thing is, overall, in average, the microbiota in the vagina and the endometrium is quite similar, but in a specific woman, there are significant differences between vagina and endometrium. And I think that in this woman is very important to analyze the endometrium and not the vagina if they are undergoing IVF, because the embryo has to implant in the endometrium, not the vagina. If you sample the vagina, you will never know what is happening in the upper reproductive tract where the embryo has to implant. So this is these differences were the ones that lead us to do this research.
Okay. Well, one more question is there. Since you were talking about the miscarriages and all during your presentation, well, do we have any data which tells us about the composition of microbiota in spontaneous abortions?
This is a very good one. We are now conducting a collaboration with Stanford University, and we are just analyzing this. So we are selecting those samples of miscarriages with euploid embryos, and our objective is to compare the microbiota of the, to analyze the microbiota of the miscarriage of euploid embryos to check whether the altered microbiota may be a factor, may be causing these miscarriages. So this is hopefully I will be able to answer this question in the future, because we are just analyzing this in a collaboration with Stanford University.
Okay. One question for the entire audience from my side and for myself too. What are the benefits of NGS microbiome? I mean to say the analysis of microbiome by NGS vis-a-vis microbial culture.
Yeah, so there are 2 main benefits. The first one is the sensitivity of the technique. Because as I mentioned before, the microbiota in the uterus is a low biomass microbiota. This means that the total amount of bacteria inside the uterine cavity is not very high, is not huge. We have a lot of more bacteria in the vagina and in the endometrium, which makes sense because the vagina is more exposed, is out in the outer part of the reproductive tract, and the endometrium is, let's say, covered by the cervix and so on. So the first thing is that NGS is much more sensitive and is able to identify the microbial DNA even if they are in a very low amount. The second one is that microbial culture is not able to culture some of the bacteria that inhabit the reproductive tract. For example, Gardnerella vaginalis is quite difficult to culture. Other types of bacteria, for example, Mycoplasma, Ureaplasma, these are very, very difficult to culture in vitro, or even they are not usually culturing in vitro. So if you are only doing microbial culture, you may miss this bacteria. You may say this sample is negative for the culture, and the problem is that you are not able to culture the Mycoplasma inside or the Gardnerella inside. And you are giving a negative result, not only because the woman does not have bacteria, it's because it's technically is not feasible to culture these bacteria. So this is another benefit. And then from the technical, let's say, level or the productive level, another benefit for NGS is that it allows to analyze a lot of samples at the same time. So for example, for microbial culture, and this is something that, I mean, is a benefit for the people that is working in the field, but you may be able to analyze different samples at the same time. So you, in microbial culture, using, for example, PCR, it is impossible to run, for example, 80 samples in a row. In doing NGS, you can do this. So the speed and the cost of the analysis is bigger.
Thanks, Emma. Well, before I allow you to, say bye to the audience, during this pandemia, if I go back to your talk, which you took over in the month of April, when COVID was at its peak in Europe and other continents. So that time, many questions were there which were unanswered. So today, Dr. Emma, if I ask you as a researcher, do you have any answers when we talk about endometrium vis-a-vis COVID-19? Would you have any message for the audience here?
Yeah, so we have been working also in COVID-19 and endometrium. As far as we have seen, there is a lot of lack of knowledge about whether COVID-19 or SARS-CoV-2 may enter the endometrium and colonize the endometrium, and this may have implications in the vertical transmission to the babies and in for example, in neonatal complications or interactogenicity. This is very important for us. We have been focused because we using single-cell RNA-seq of the endometrium for another project, we had the advantage of have a very big collection of bioinformatic data about endometrial single-cell RNA-seq. And we have sent these results, we are preparing these results for publication. What we have seen is that the port of entry of SARS-CoV-2 in the endometrium, which is this protein ACE2, and the proteases TMPRSS2, cathepsin B, cathepsin L, they are very low expressed in the endometrium. This means that the possibility that the endometrium is colonized by SARS-CoV-2 is very low. I won't say it is impossible because it's very early to make this type of assumptions and to make these conclusions. But what we have seen is that the probability or the risk of having infection in the endometrium is quite low. It's below 1%. And this is what we have been working so far. As I said, we are also progressing with these types of study, and as far as the pandemic is still on, we will continue working in the field, and we are also trying to modelize endometrial infection by SARS-CoV-2 in vitro to explore more mechanistic studies. But this is what I can tell you right now. We have been doing some small improvements in our knowledge, but still working hard on this sense.
Thank you, Dr. Emma. It was a pleasure having you here with us. Thank you very much for taking up all the questions and replying to all the queries in the best possible manner. Thank you very much. And I would like to, thanks, Dr. Emma. And I would like to thank the audience for being here with us. My gratitude to all of you on behalf of Igenomix family for sparing your time for this wonderful presentation. And before we wrap it up, I request you not to forget to attend the upcoming and the last episode of this webinar series, which is on miscarriages, the latest updates by Dr. Nasser Al-Asmar. So please be there with us. And just a reminder for GIF Symposia, which is there on 3rd of September. You will be receiving furthermore notifications about this GIF Symposia. So please register yourself. Thank you very much. And thanks, Dr. Emma. It was wonderful having you here with us this evening.
Bye-bye.
Bye.